Got a great panel here. So let me turn it immediately to you in the audience, and hopefully you have some questions, and if not, I'll exercise my role as moderator and try to stimulate the discussion. But please, I'm sure you have some discussions for the panelists. Yes, sir.
Well, a research scientist at the Northeastern University and also research fellow at the University of Glasgow. I want to ask, I share with you, especially with the presentation of Paul, about this set of collection of compounds that can be easily tested to for pharmaceutical companies that work in this field. I'm a computational chemist, and sometimes I feel the challenge sometimes to get the compounds I want, because, for example, they collect a lot of compounds, but they are not available. Like, for example, for chemical, a very big list of compounds, but they are not like ready to test. So sometimes it would be good to actually maintain this database that consists of compounds that are easy to test and are easily accessible, not that the companies sometimes keep it for themselves. So I would like to share this.
Thanks, and just for clarity, that's to test in a physical sense in a screen. Yeah. I mean, that's something we could look at it. It's actually quite difficult because it's a dynamically changing situation that, compounds are commercially available, then the stocks have run out. So it's something that we find a challenge, but thanks for the feedback. We'll see what we can do.
Thank you. Please go ahead, John.
A tiny comment from the Kemble end. So there's another resource behind Kemble called Unichem, which contains about 100 million structures, about 80 million. And a large number of those are accessible, so it's easy to do mapping of available to Kemble. You can actually filter Kemble for compounds that you can physically order. It's about 8 to 10% of everything in Kemble you can get hands off. But the devil's in the detail. You've got things like salts, stereoisomers, all sorts of things. But yeah, I think it's an important point. The other thing we've started to do is, I guess, think about the classifying sets of compounds as to whether they're available, whether they're approved, whether they're reasonably synthesizable, and so forth, or real just prophetic virtual compounds.
In the case of GSK, all the compounds for which we release data, we make them physically available. So, straight and simple.
Thank you.
Cynthia Dugan, US, in Singapore. Yes, do any of these databases have toxicology information? That seems to be a rate limiting step to get into first in human. So it'd be awfully nice some information about that in the public domain.
You're the most likely.
Yes.
Sorry. So we store a lot of, well, one of the issues, I guess, with toxicology is you've got to have reasonable exposure of your compound in an in vivo setting. And so getting the exposure, you've got to do a lot of lead optimization and med chem to get there. So the number of compounds for which you've got reasonable tox data is very small. We've snaffled up what we can. There's some great projects out there, things like TG Gates, the NIEHS in the States have got a lovely data set of in vitro and animal-based tox data as well. So there's less there. I think it's very valuable. I think one of the other data sources that would be that's cryptic or locked away at the moment is a lot of the regulatory submissions of matters of public fact. So depositions to NDA applications, submissions to the EMEA. There's a lot of molecular data there, but it's just not accessible. So we're looking seriously about trying to dig that out and make that open as well.
And let me add to that. I would say one of the things that's interesting about the day, and certainly the presentations that we heard after lunch, is the wide range of models that are being developed depending on the type of institution and what aspect of the problem we're trying to address. So in the case of WIPO research, I would say certainly in terms of our database, which I'm thinking of Michael Pilastri's comment about Excel not being a data set, in a way, the WIPO research database is not really a database as such. It's more a catalog of assets. But so we, for example, do not list that kind of information. But what we offer is by helping providers and users establish each other's bona fides through the partnership hub, we make it easier for a company or some other institution that's developed a compound to give that information to a researcher. And again, you all are research scientists, so you know this better than I do. Often the value in science is knowing, what road not to go down. And there are several of our collaborations or communications that have been facilitated through WIPO research where research had an idea for a certain path to pursue, talked to somebody else who owned a compound or a similar compound, had tried that, and said, no, that's a dead end. So it saves months and months of time if you know in advance what the answer is. And that's, again, one way that we provide that information is through the direct one-on-one collaborations or communications. So even though the information isn't publicly available just by going on the website, it is accessible.
Okay. This is Roxanne Kemsey from Nature Medicine, and this question came to mind particularly in the talks that John and Michael gave, but I wonder if anyone else has things to add. My question is really how all of you are working with the technical side of this when you're building databases or platforms to facilitate these collaborations and sharing of data. And I was just curious to know what kind of challenges you might encounter in that kind of technical aspect of these things that you're building.
Mine is the least technically required. It's not, as I mentioned, it's all chemist proof. So you basically just upload data and you look at data. The issue for us, John's going to have a lot more to do, but to talk about probably than me, but one of the things for me, just maintaining my own lab's database, right? So just the dozen or so people is curating the data. So when people upload or people provide data, I always have to clean the spreadsheet before I can upload it. Or I get structures to register and they have some error. They don't have the salt included and stuff, and I end up manually curating that. That's going to be something that is not easily, growth is going to make that a lot more work, such that it's not trivial. It's probably going to require a person to do it, someone to maintain it and make sure that the database is actually useful and not full of errors.
Dr. Holtz?
Well,
Yeah.
maybe I can say about it as well, because I'm from CDD, which is providing the world, and I worked for software companies for many years. So scalability is usually a huge problem. So you start to promote your tools and ask people, invite people to participate, and then the software doesn't perform, then of course people will turn away. I find compliance and the culture of compliance is of immense importance. So if you're asking people to share data with each other, then that requires a level of trust, and that usually is established through a culture of compliance. So you stick to the rules, you do share, I think, everything that Mike mentioned in his talk as well. So somehow making the usability of the software help compliance and the culture in a collaboration. So it's performance as well as usability for the software makers. And then, of course, it's the security issues are huge. So how do I establish trust as a software maker that people feel, yes, I can deposit my data here and I'm safe. My data is relatively safe, especially with what was in the media recently, right? Especially in Europe, a lot of people are saying to me, well, what about how safe is it? Who else is listening? Who else is reading my data? And that's something, of course, that you can't answer, but you can take measures, and the NIH does have certification procedures as well. So there are help to prove compliance. But if you ask me personally, having worked in software for a long time, compliance usually breaks down at the personal level. So in my opinion, having a culture, promoting a culture where people will comply to the rules and trust the environment usability. So if the more easy it is to use, the more people will do what they are supposed to be doing. And I think those are the challenges from the technical and from the software maker point of view.
Thank you.
So a couple of comments on the technical aspects of the data itself. So I guess we've probably all seen some papers that have shown that in particular in vivo compound-based results are very irreproducible, at least those that are reported in the literature, like 60% can't be reproduced or something. And people, yeah, they say, is this fraud, or is it just poor experimentation or whatever? I think part of it is our current process of reporting results doesn't allow enough specification in the journals for people to reproduce the experiment. And there's always pressure on space and so forth, and even supplementary material now you can't put everything in. So I'm really excited about some of the things that are coming out of say, Stefan Schurer's lab in the States. He's developing this bioassay ontology, which gives a really very sort of concise framework to define assays, and hopefully will lead to better reproducibility. We see this with even things like log-P assay. You can do this in a lot of different ways, and so the same compound in a different lab will give rise to different endpoints. We look at the differences of the same compound, nominally the same assay, across this cascade of biochemical, through cell-based, through to in vivo data. And by the time you're up at the in vivo level, you often get those response curves that are 100-fold offset, probably because of minor differences in the animal strains or the food or whether they were dealt with by men or ladies or whatever in the experiment. So there's a lot of variables that are there, and I think the more we capture those, the more we can reproduce and extend the work of others.
Thank you. Yes, sir.
It's working? Okay. Hi, my name is Paul Horrocks from Keele University. Something that struck me when I was listening to today's presentation is about 15 years ago I was heavily involved in the Malaria Genome Project, and we were facing the same sort of questions. How do we share and use the information we have? And so, we've gone into the bio. We've got all. Is that working? Okay, usually I just shout, it's easier. But we have a fantastic tool now, a bioinformatic tool that we share, and it's growing and it's getting larger. But one thing that's striking us now is it's about sustainability of that resource. Now, I mean, John certainly touched on that today when he was talking about the renewal of his grant. There's lots of innovations here. Getting them together and actually having that sustainability for the next 20, 50, 20, 25 years, which is realistically what we're going to need. What's your views on how we can do that? And my apologies for cutting in and out. I usually shout, it's easy.
Well, that's a $1 million question.
No, actually you need more than $1 million.
Yes.
Well, that's
I'm dating myself, yes.
It keeps me up at night actually thinking about this, right? I mean, it took us 3 months to raise $25,000 with crowdfunding, so that's not going to work, right? I mean, for our perspective, for our model, we're hoping that we can do this as a proof of concept experiment with the seed money and then convince a funder. By the way, I don't know how you would get NIH money for this. Like, they don't generally do that, and I'm pretty sure based on my conversations with the Gates Foundation, they don't like to fund this kind of stuff either. So I don't know the answer to your question. Thoughts are welcome.
Yeah.
And I'll be passing the hat later. Yeah.
Others?
For me, I guess the backstop of open data is that there will be copies. So if Kemble wasn't funded, there would be a static snapshot of the database that would be on thousands of servers around the world, and it would be, someone else could pick it up if the funding environment changed. So I think we need to think about robust mechanisms. I think in Europe there's a number of infrastructures for data, data funding security, sustainability. I guess the one closest to my heart is this Elixir project, which is trying to bring together European infrastructure funding for life science data. But it's a very complicated process. Everyone says it's a good idea, but getting real cash on the table is very tricky. At the end of the day, people talk about hybrid models of the commercial sector should pay for things and it should be open to academics. I don't think that's really tenable. I think you just need transparency, equivalence of access for everyone, and try and really build sustainability on the back of that.
Is that the idea that it has to be a common base to the data so that you can utilize one of these GUS type mechanisms that is searched? Because if you can't search the information, then it's just a static resource.
Static resources, it's very difficult to utilize those in an exploratory sort of scientific approach. It has to be dynamic, evolving, and searchable. And I think that's always going to be the challenge.
I think more importantly, it needs to be distributable. You need to take your own copy. You need to be able to integrate your own data against it without restriction, and so forth. And I think the aspiration of building a big monolithic system that does everything perfectly is just not sustainable. For Kemble, we love the data, we put an interface on it, but the interface isn't going to be to support compound optimization or whatever. It's going to be serving the data to people who know how to use it better than we do.
You can claim that the database is the cheap part of the story.
With sustainability, I'm looking a bit as an outsider. I'm not a drug discovery researcher, but what I'm seeing is that these collaborative models that are being tested and established in the neglected disease area, I would hope, but I don't know, maybe the people in pharma can answer that. Is that going to be something that's going to be picked up by pharma as a model for drug discovery in the future, and that will then establish hopefully best practices, format exchanges, and things that will help people to move forward and still look back in 20 years' time and say, wow, we are still using data that we generated 20 years ago in those first projects. That would be my hope.
So there is an appetite within industry to extend some of the practices that we have seen today to other therapy areas. I can respond certainly for GSK, but I can see that in other companies that the same trend is developing. So I think that for the time being, as long as the rest of the therapy areas have not embraced that, we have an opportunity to explore, and I think we'll have the support from not all, but maybe some funding agencies and some pharmaceutical companies. Maybe it will not be enough, but we have to try.
Thank you. Other questions and comments?
Hello, I'm Jean-Yves Trosset from Sub Biotech in Paris. You mentioned about missing data and missing compounds which are inactive compounds. Is there any effort to collect like inactive to make a star, at least to be able to do repurposing studies and much easily?
The biggest data repository that has this, Kemble has this to some extent, no? PubChem does, right? So all the high-throughput screening data that got screened at the NIH screening centers, for example, all those negative data there. The ones I'm most concerned about are the medchem stories that are never told, right? I've had a paper rejected by a whole bunch of journals, and it's because we didn't get a 1 nanomolar compound that's super selective, right?
Yeah.
We got to tell the story, because someone may do the same thing and waste all kinds of time. So we need to have, as a community, a way to get that data to the public.
Yeah, exactly. Yeah.
I feel embarrassed pushing GSK's name in this setting. It sounds like we're best friends or something, but we're not. We hate each other, really.
It's not true.
One of the things that's good is the literature doesn't allow the reporting of a bunch of JMed Chem or whatever, BMCL, whatever the journal wouldn't allow the reporting of a whole bunch of boring inactives. So GSK have committed as part of this Open Facts project on some of their SAR disclosures of some of the lead series, to report a larger set of compounds of the bioactivity directly deposited into Kemble, and you can see in some of the supplementary data sets that we have this. So typically in a paper you'd get 20 or 30 active compounds that tell a bit of a contrived story between the lead and the clinical candidate. And that's nice, it makes sense. But what you really want to know is these inactives and some of the side tracks that weren't explored, and have the data for those. And I think it would be great if more companies did that, and certainly within Europe and in some of these, in particular these IMI projects, there is now this appetite to share some of this extra shell of contextual data around the key scientific breakthroughs.
Because I do understand that companies might not want to share all their data, but at least to have a rule like in the SGC, they make a rule like we can establish similar rules. For example, at least 1% of inactives should be published together with a list of actives. Or there are 1%, at least few compounds.
I love that idea.
Yeah.
But I think it's actually very difficult at times to define what is active, and then what is your so an assay dynamic range often isn't very large. A lot of assays have only really got, say, 10 to the 2 potency range, unless you really work on your assay optimization. So what would be an inactive compound?
Yes, true,
is it 10
true.
micromolar, 100, 4 millimolar? It's difficult. It's not as simple as it first appears.
And I think in fairness, if you consider the average farmer is running tens or hundreds of high throughput screens a year,
1% plus 1%
quite
plus
quickly
1%.
their IP's gone. So I think we have to live with that.
I think it's a really interesting point, and it might be worth commenting as well. The Royal Society of Chemistry, for example, I know are working at initiatives with micro-publishing, where perhaps you don't have the data set for full paper, but those smaller sets of active, inactive, you could get out quickly. I mean, Ali from the RSC is in the fourth row. I don't know if you want to add anything, Ali?
Well, I just to comment that the Royal Society of Chemistry supported micro-publication and as well the publications of negative data. And I think that we understand that there is a lot of information that is needed and is required by the community, not only in the neglected diseases area, but in all areas. So this is something that we are really active now.
I mean, it makes a lot of sense, right? You don't want people to just send data that's just bad, right? It needs to be scientifically robust and controlled. I mean, it has to be good data. It's just the compound didn't work in your hypothesis, right? There's got to be a null hypothesis way, some way to publish null hypotheses.
Fascinating. All right. Well, thank you very much. Thanks to our very distinguished panel. It was a very informative afternoon. Thank you for your time. Thank you for your attention.
Distinguished.
Thank you very much.